Alright, so let's get started with the subject for today's revision webinar. This is going to be on the topic of infection and response for GCSE Biology students. So let's have a look at what we're going to be covering today before we get started. So these are the topics. Now, as per usual for these revision webinars, we're going to be covering the topics not necessarily in this order, but to whatever extent possible, we're going to be covering them in the order in which you voted for them, those of you in attendance on the private resources page. Now, again, if you need to find the resources page, you need to have a look at the very first page of the PDF version of this document that you received when you were booking your tickets, and then click on this link here. You do need to be logged in to get to that page. Many of you have already accessed that page because you voted for the topic order that we're going to be covering in the first hour and also in the second hour, should you wish to do so. You also, many of you voted for the exam questions from the exam style questions pack that you received in terms of which ones you found most challenging. Now, should you all choose to go into the exam questions in the second hour, we will be covering those exam questions also in the order in which they received votes. Now, in the second 5-minute break after the second hour, again, you're going to be able to vote for whether or not you'd like me to continue for an extra 30 minutes, and if so, whether you'd like me to continue for that 30 minutes on topic teaching of the topics for this webinar, or whether you'd like me to continue by going into the exam questions in more depth. Now, whether or not you choose for me to cover the exam questions, I will be uploading the complete mark scheme to those exam questions to that private resources page within about 48 hours of the end of this event. Excuse me, still got a bit of a sore throat. All right, so let's get started then. So I think a good place to start for this topic would be to look at what we mean by the term pathogen in general and how pathogens cause disease. Again, in general, we're going to be looking at specific diseases and specific pathogens in a little bit more depth a little bit later on. So diseases can be caused by microorganisms, so certain types of bacteria, viruses. Now, these are commonly called germs, although the word germ is a little bit not particularly rigorous scientific language. When these types of organisms, these disease-causing microorganisms enter your body, they can then cause various symptoms of various diseases. So microorganisms which can cause disease are said to be pathogenic, and those types of organisms are typically referred to as pathogens. Now, because of the actions of the various barriers to infection that you have in your body, and also because of the ability of your immune system to fight off these infections, diseases from these types of pathogenic organisms are more likely to occur if large numbers of them get inside your body, get past those exterior defenses of your body, such as, for example, your skin and so on. And so this is more likely to happen as a result of unhygienic conditions or as a result of contact with infected people. So pathogens in general will cause the symptoms of a disease in various ways, but in general they make you feel ill by doing damage to body tissues and also by producing toxins or poisons, which can then cause the kinds of symptoms that you will experience as a result of infectious illnesses, things like fever, high temperature, vomiting, headaches, and those sorts of symptoms. Now, typically, these kinds of symptoms don't appear immediately after infection. There's an interval called the incubation period, which is the time taken between the actual infection by these organisms, the event where they enter your body, and the time between that event and when the symptoms actually start to be experienced by you. Now, it's during this incubation period when the microorganisms, the pathogens, are rapidly multiplying inside your body. And so in the same way that you would incubate a bacterium on a petri dish or some other culture media, in a similar manner, when these organisms get inside your body, there is an incubation period during which they multiply rapidly inside your body. So let's start by looking at bacterial pathogens. Now, bacteria, as we mentioned before in the first GCSE Biology webinar on the subject of cell biology, these are prokaryotic cells, and they're typically much smaller than cells of eukaryotic organisms like ourselves. And so they typically have a cell size between 0.0005 millimeters and 0.005 millimeters. And they also share particular features, some of which are common to all bacteria, some of which are found only in certain types of bacteria. And so we have, for example, the slime capsule, which prevents desiccation under dry conditions. We typically also have ribosomes, which are smaller than the ones found in eukaryotic organisms. And we also have these, sometimes have these small loops of DNA called plasmids, which can be transferred between individuals, and these are implicated in the spread of antibiotic resistance. When one bacterium develops antibiotic resistance, it can then pass on that trait by passing the gene for that antibiotic resistance from itself to some other bacterium. And so this is done by means of these small loops of DNA called plasmids. We also have this circular loop of DNA, which is the bacterial chromosome. And bacteria also have a cell wall, which is made up of a different type of material than the cell walls found in plant cells or fungal cells. And some bacteria also have a flagellum or multiple flagellae, which are these long thread-like projections which emerge from one end of the bacterium and actually rotate like a propeller to propel that bacterium through fluids. So bacterial cells, like eukaryotic cells, unlike eukaryotic cells, I should say, typically do not have internal membrane-bound organelles, and as such they lack a nucleus as well. And so they also have a tough protective cell wall made from a very different material to that found in plant cell walls. The bacterial cell wall is made up of a glycoprotein called murein. Now, as with all organisms, whether prokaryotic or eukaryotic, DNA is the carrier of genetic information. So tetanus is caused by a bacterium, Clostridium tetani, which causes the symptoms of tetanus by releasing a bacterial toxin, which actually is a neurotoxin. It causes poisoning of nerve cells. And so we see here that this infected person has some of the typical symptoms of tetanus, where the man's head is twisted and locked to one side due to the sustained muscular contraction of the neck muscles. So the word tetanus in medicine is a word used to describe sustained and uncontrolled muscular contraction. And so an old name for tetanus is lockjaw, because the muscles of the jaw and the face are also contracted in a sustained contraction, and so this makes eating and drinking in a normal manner impossible to these infected individuals. Now, there are vaccines available against tetanus, and an efficient antitoxin has also been developed to ameliorate the effects of the neurotoxin which the pathogen which causes this disease releases. So tetanus is an example of an illness which is caused by a bacterial pathogen, but a pathogenic bacteria can cause a variety of different illnesses, and these include things like food poisoning, boils, whooping cough, leprosy, diphtheria, cholera, also dental decay and venereal diseases. Now, under favourable conditions, bacteria can reproduce by a form of asexual reproduction called binary fission, which resembles but is not quite the same as mitosis in eukaryotic cells. Now, they can reproduce under favourable conditions roughly every 20 to 30 minutes, which means that one bacterium can become 16 million bacteria in just 24 hours. Now, some bacteria can produce spores as part of their life cycle, and so this diagram illustrates the way in which these types of sporulating bacilli can reproduce themselves. Now, the function of the spore in this phase of the bacterial life cycle is to allow the bacterium to survive under conditions where there are kind of environmental hazards to that bacterium, things like boiling water, disinfectants, and they can also survive drying out and desiccation in this manner as well. So anthrax is a particular type of sporulating bacterium which shows this life cycle. So to give a sense of just how rapidly bacteria can multiply by asexual reproduction, if we imagine this is a single bacterium and we are counting the number of bacteria in this sample versus time, if we start timing, we can see that the rate of bacterial multiplication is not linear, it is exponential, because again, 1 becomes 2, 2 becomes 4, 4 becomes 8, and so on. And so In this manner, if you were to wipe down a surface with an antibacterial agent and killed off all except for one of those bacteria, very rapidly that single bacterium could then go on to reproduce to form a colony of a large number of bacteria on that same surface within a relatively short period of time. And so in this way, a large population of bacteria is built up very, very quickly. So let's take a look at viruses now. Now, viruses are acellular particles. They are not made up of cells. Now, going back to that first GCSE Biology revision webinar earlier on in this series, we know that the smallest living unit of matter in biology is defined as a cell. And so viruses as such are not considered to be living organisms because they're not made up of cells. They're in fact much smaller than cells, and also only really show one of the characteristics of living organisms, that one being reproduction. They don't respire, they don't grow, they don't do any of the other 7 characteristics of living things. They simply reproduce, and so they are acellular reproducing particles. Now, these are so small that a row of 1 million average-sized viruses would be only about 5 millimeters long. And so again, you need to have some sense of the relative sizes of some of these organisms. And so here we have an E. coli bacterium, which is a kind of average-sized bacterium. Now, in comparison to that, we have also a smaller type of bacterium, the Diplococcus, responsible for pneumonia. And you see that bacteria can actually have relatively large range of sizes. Now, again, in comparison to eukaryotic cells like our own, bacteria are much smaller, anywhere from 20 to 100 times smaller than the average eukaryotic cell. Now, in comparison to even the smallest eukaryotic cells, viruses can become extremely small. So we see here poliovirus is a tiny fraction of the size of even one of the smaller bacteria. So all viruses, by definition, are parasites. They cause many diseases of not just animals, but plants, and they can even infect bacteria. So here we can see a diagram of the HIV virus. This infects humans, but you can also have viruses which infect plants, like the tobacco mosaic virus. You can even have bacteriophage viruses which infect bacteria. So viral diseases of humans include things like the common cold, which is actually caused by a variety of different viruses, but also influenza, smallpox, hepatitis, polio, and also rabies. So some viruses are even implicated in the development of cancer as well. And so there are viruses like Epstein-Barr virus and so on, which have been shown to increase the risk of certain types of cancer. Now, viruses, again, are not cells. They do have DNA within them, and this DNA is enclosed in an envelope of protein and sometimes lipids as well. Now, unlike other living diseases, viruses don't feed, they don't grow, they don't move, they don't respire. All they do is reproduce by inserting their own genetic material, their DNA or sometimes RNA, if you're dealing with an RNA virus, into the host cell. And then the host cell will then replicate that virus. It will copy that virus by making use of the genetic instructions on the viral DNA that's been injected into it. And so viruses effectively live by hijacking cells and turning them into virus factories. So the life cycle of the virus involves the virus attaching itself to the cell membrane of the host cell and injecting its DNA into the cell's cytoplasm. The virus DNA then causes the host cell's DNA and other organelles to make hundreds of replicas of that virus. Now, after as little as 30 minutes, the host cell containing all of these new viruses bursts open, and then that releases those new viruses into the surrounding extracellular medium where they can go on to infect other cells. And in fact, this is the mechanism by which viruses often will cause their symptoms in human beings. It's by this damaging of tissues due to the viruses infecting and bursting those cells open. So here we have again the bacteriophage virus. Now, bacteriophage viruses will attack and kill bacteria in the same manner that viruses that infect and kill eukaryotic cells do. So now, different viruses will have very different morphologies, very different shapes. In the case of a bacteriophage virus, it kind of looks a little bit like the moon lander. It's got a so-called head, which contains a strand of DNA. Now, the DNA contains all of the genetic instructions necessary for making all of the components of each virus. And so the viral DNA then uses the bacterial DNA and the internal machinery inside that bacterial cell to build more viruses. And when these are released, they can then go out and infect more bacteria. So the same process takes place when pathogenic viruses infect the cells of human beings and plants. So here we can see the head group containing the DNA. So the head itself is composed of proteins which encapsulate the DNA of the virus. We also have the tail of the virus, also made of different kinds of protein. And inside the head we have the DNA strand for the virus. So to help you understand the life cycle of a virus, this is a kind of animation which shows the stages of the virus's life cycle. We start with the virus attaching to the cell membrane of the bacterial cell and then injecting its own DNA into the cell. That DNA then combines with the bacterial chromosome. And so the DNA in general of organisms is essentially a series of instructions that tells the cell how to build proteins. And so in the case of the viral DNA, it instructs the ribosomes and other cellular machinery inside the bacterial cell to assemble the components, the protein components of more viruses. We also have the replication, the copying of the viral DNA again and again as well, until eventually these can be assembled into new viruses which then burst the cell open, and in so doing obviously the bacterium is destroyed, and this allows these new viruses to infect surrounding bacterial cells. And so this is very much a similar process that occurs for the viruses which infect human cells as well. So here we have a parasitic species of protozoan. Now, protozoa are eukaryotic unicellular organisms. An example of one of these is amoeba. Now, a particular type of protozoan called Plasmodium is responsible for the disease malaria in humans. Now, this type of disease is carried by an animal vector. So the word vector means a carrier. And so in this case, it's the mosquito which carries the malarial parasite, the Plasmodium. And so the mosquito-borne parasite is transmitted to humans when the female of this species of mosquito, the Anopheles mosquito, infects a human by taking a meal of blood from the human skin. And so the Plasmodium, the protozoan Plasmodium, which causes the illness malaria, is carried in the mouthparts of the Anopheles mosquito, and then this goes into the blood of the human host when the mosquito bites and feeds on the human's blood. Now, the health and economic implications of malaria are huge. This parasite kills more people each year than any other microorganism in the world, and this is despite huge efforts to eradicate this organism altogether. So there are other types of pathogens, other types of protozoa. So this one here, Histolytica, is a protozoan parasite which causes amoebic dysentery. We also have other malarial or other protozoan parasites, and these cause other illnesses such as sleeping sickness. And so here we can see that this particular protozoan species also has a flagellum. So flagellae are not unique to bacteria. They can also be found in eukaryotic unicellular organisms such as protozoa. Now, there are fungal illnesses as well. Now, ringworm is a fungal infection of the skin, and it spreads outwards, forming a circular red scaly area. Athlete's foot, which is an infection between the toes, is caused by a closely related species of fungus. So here we can see a diagram showing the branched strands which emerge from these fungal organisms. These are called hyphae. And these emerge from the spores of the fungus under the correct conditions. Now, dandruff, which is not the same as dry scalp, dandruff is also caused by fungal infection. So, Infectious illnesses can be caused by a variety of different microorganisms, but they can also be spread by a number of mechanisms. So diseases can spread from person to person, and these types of diseases are said to be contagious. So let's have a look at some of the ways in which contagious infectious diseases can be spread. So firstly, there is direct human contact. So diseases like these can be spread by directly touching infected people or objects that have been handled by infected people. So examples of communicable diseases that can be spread by direct contact between individuals are things like smallpox and measles. So the main contact points are shown in this diagram here. We touch objects and each other with our hands typically, by shaking hands and so on. So of all of the areas of the hands, it's actually the fingertips which are most responsible for picking up pathogens and spreading them to other people. And so obviously a good practice is to wash the hands regularly, and this is obviously one method that we can use to avoid the spread of infection either from other people or from touching objects that have been touched by other people. So when you cough or sneeze, you can spread infectious microorganisms in droplets of moisture, as is shown by this photograph of this person sneezing. So contagious diseases which can be spread in this way include bacterial and viral diseases. So you've got flu, pneumonia, whooping cough, tuberculosis, and also diphtheria. So infection can be contracted by either directly inhaling the droplets or touching surfaces onto which those droplets have fallen. Now, another mechanism by which communicable diseases can be spread is through contaminated food. So food can be infected with microorganisms from either dirty hands or contaminants such as sewage, insects, birds, and other animals. So typhoid, cholera, dysentery, and most types of food poisoning are spread in this manner. So in this photograph, we can see one of these methods of the spread of these microorganisms. So flies will salivate and leave their feces on food, and so it's important to keep flies away from food. We've mentioned already one animal vector, so insects and other animal vectors which spread diseases. So the word vector in general, in this context of communicable disease, is an organism such as an insect or other animals which can themselves spread diseases to other organisms. And so we've looked at a couple of examples, the fly and also the mosquito. Now, yellow fever is another example of a microorganism-caused illness which can be spread by mosquitoes. Obviously, malaria is the big one. So mosquitoes have piercing mouthparts and can suck blood from the host, but the mosquito also pumps saliva into the wound, and the saliva contains an anti-clotting agent to prevent the blood from clotting to allow the mosquito to continue to feed. And so there are other illnesses. We've mentioned yellow fever, but there's also elephantiasis, which can also be caused by these disease-carrying mosquitoes. Now, malaria can be fatal, but usually causes fever, headaches, and vomiting. And again, this is caused by a protist. Elephantiasis is caused by tiny roundworms, which can block lymph vessels, causing the limbs to spread. And yellow fever can also be fatal, and it's caused by a virus which actually damages liver tissues. And so here we can see one of these microorganisms with the various parts here, or rather the mouth parts of the mosquito, showing the way in which the mosquito uses its mouth parts to penetrate the skin. Now, ticks are arachnids. They belong to the same group of organisms that spiders belong to. And so these are actually parasites of vertebrate organisms such as ourselves. So they use hooked mouthparts to penetrate skin and suck the blood after, again, injecting anticoagulants, chemicals that prevent the clotting of blood. Now, these can cause serious blood loss and also paralysis in their hosts. The ticks will then drop off from their host when they're full of blood. So here we can see the brown dog tick. These can spread a large variety of diseases to domestic and also wild dogs, and also other animals such as cattle and sheep. So this species of tick can also cause typhus in humans. Now, fleas have a body which is flattened from one side to the other, and feet with tiny claws. Now this lets them move easily between the hairs of mammals. So here we can see the pestis bacterium. This causes bubonic plague, and this can be carried by rats and fleas like these. So the fleas also suck blood, and it's also in this manner that they can spread these bacteria. Now, at the time of the plague in 1665, nobody had a correct understanding of the causes of communicable disease, and this obviously led to many, many deaths before eventually the spread of the plague was stopped. So we've talked about a number of the causes of infectious illnesses. We've talked about the ways in which these infectious illnesses can be spread. Let's now move on to look at the ways in which living organisms can control the spread of disease and prevent infection naturally. Okay, so before we go any further, one of the things that many of you currently in attendance voted for me to cover was the idea of immunity and the ways in which immunity are categorized. Now, the categorization of different mechanisms of immunity can cause a lot of students some issues. Now, in order to kind of better understand that, let's start by looking at the various kinds of immunity which can be categorized in various ways. So before I go any further then, let me ask the question to those of you in attendance. This should pop up on your screen. If you don't see it pop up on your screen, you can go ahead and use the chat box to answer this question. So first of all, what does the word immunity actually mean? How would you define the term immunity if you were asked to do so in an exam? And the secondary question that I'd like to ask at this point is, if you had to categorize immunity into two general types, how would you do so? So I'm going to pause for a moment here and give the attendees time to answer this question. All right, so it looks like across the 5 groups of students, most of you managed to answer the first part of the question correctly. There seems to be quite a lot of variation in the answer to the second part of the question about categorizing immunity, and don't panic, that's fine, because there's actually a couple of different ways we can categorize immunity. So we can categorize immunity as either being innate immunity or natural immunity as it's sometimes called, or we can categorize immunity as being acquired or artificial. That's two different ways of categorizing immunity in terms of where the immunity comes from, but there's also a couple of other ways of describing immunity, as we'll see as well. So let's define immunity first then. So immunity can be defined as the ability of an organism to resist infection. And in general, in terms of the first type of categorization, which I'm going to go through, there's two kinds of innate immunity. There is non-specific immunity, and there is specific immunity. Now, non-specific or innate immunity is provided by two main methods, and both of these kill most pathogenic microorganisms. Now, these are called non-specific because they don't target a particular pathogen, a particular microorganism, particular bacterium or virus or anything like that. These are general barriers which will kill most pathogens, regardless of what type of pathogen that you're talking about. And so these will include things like physical and chemical barriers and also phagocytosis. So we're going to look at these in a little bit more depth shortly, but this is the general way of categorizing, or rather this is how we would define non-specific immunity. Now, in contrast, specific immunity is largely to do with the actions of the immune system. So specific immunity is the targeting of a particular pathogen which has been detected by your immune system, and this is where the body takes steps to protect you against a specific disease-causing microbe. And the specific immune response can be active, or which has a long-lasting effect. It can also be passive, which has a short-lived effect. Now, this subject of immunity in the immune system is extremely complicated, and at GCSE level, we're simplifying things somewhat in these ways of categorization. Those of you who go on to study advanced level biology will start to understand that actually this is a bit of a simplification, but for GCSE purposes, if you're aiming for the top grades, this is absolutely fine. So let's take a look first of all at non-specific immunity and the ways in which this is achieved. So if we had to define non-specific immunity, this is the body's first line of defense against infection. And firstly, it does this by presenting several physical and chemical barriers which prevent these microorganisms which can cause disease, these pathogens, from getting into your body tissues and causing a problem in the first place. So these include the skin, the eyes, the nose, the trachea and the airways themselves, the stomach, and these are the kinds of outer barriers to infection. Now you might be thinking, well, the trachea and the airways, these are internal parts of the body, as is the stomach. That's true, but the airways and also the alimentary canal, the digestive system, are effectively continuous with the outside of your body because the air carries a lot of microorganisms, and when you breathe air in to your trachea and your bronchi and your bronchioles and eventually into your lungs, into the alveolar air spaces, that air is effectively continuous with the air in the outside world. And so essentially, even though the trachea, the inner lining of your airways, is within your body, it is essentially an external barrier because it faces the air that comes in from the outside. And the stomach is very similar to that because when you swallow your food, essentially your entire digestive system is a tube which at one end and at the other is continuous with your external body surface, and the food that you're ingesting contains microorganisms which have come from the outside world, from outside of your body. So the skin, the eardrums, the linings of the nose and the respiratory system, these provide a physical barrier which has to be breached by pathogens in order to cause infection. So the cilia and mucus in the respiratory system add to the barriers that the microbes face. Now in the stomach, we also have stomach acid. Now the stomach acid has two main functions, the first of which is to kill pathogens and other microorganisms that have been swallowed in your food. But also there are other factors inside the body which can also limit the rate of microbial growth, and these include temperature and oxygen levels. So let's take a look at some of these physical and chemical barriers to infection a little bit more closely. Let's start with the eyes here. Now eyes are protected by tear glands and eyelashes and also by the blink reflex. So here we have the white tissue layer of the outside of the eyeball. This is called the sclera. And here we have the eyelashes. Now the eyelashes protect from dust particles, and the dust particles that you find floating around in the atmosphere are heavily colonized by various types of microorganisms. Here we have the pupil, and this controls the amount of light entering the eye. We also have the iris, which is a layer of muscles which controls the size of the pupils. Now, it's really the tear ducts and the tear glands which are very important in preventing the infection of the eyes, because the eye surface itself is moist. It has to be moist to facilitate blinking and also to protect the tissues, which are very delicate, from being infected and colonized by pathogenic organisms. So the tears contain a mild antiseptic, and this then washes the outer surface of the eye, called the conjunctiva, when we blink. The blink reflex itself, which is it is a reflex, so it is an unconscious automatic action that occurs when the surface of the eye is touched. Now the blink reflex protects us against physical damage to the eye, and if the conjunctiva's surface was to be damaged, that could then open up a route through which external pathogenic organisms could invade your body tissues. And so the blink reflex and the tears are the main protective mechanisms that prevent the entry of microorganisms into your body through the very delicate eye surfaces. Now the outer layer of the skin is covered with a layer of dead skin cells, and this also presents a barrier to infection. So here we can see a section through the skin showing the various layers of the skin. We have the sebaceous glands, which produce an oily substance called sebum. So underneath this layer of dead cells is a layer of growing, dividing cells which replace the layers of dead skin cells as they flake off. Now, the outer layer of dead cells is replaced every 3 days, and it's in fact the dead skin cells which accounts for about 90% of household dust. excuse me, frog in my throat. The hair and the layer of dead skin cells are kept subtle, waterproof, and mildly antiseptic by that oily fluid called sebum, which is produced by the sebaceous glands. Now, this makes the skin a waterproof, germ-proof, and self-repairing barrier which prevents the entry of dirt and also disease-causing pathogenic microorganisms into your body. So again, here we can see the cornified layer, the stratified layer of layers of dead skin cells. Now this again is a layer that stops germs and dirt from entering the body. Here we can see the germinal layer underneath that layer of dead skin cells. This is the layer of rapidly dividing living cells, which eventually replace these layers of dead skin cells as they flake away. Here we can see the hair follicle, the sebaceous gland, and we can also see the blood vessels in the hair root. Now, the other important aspect of the skin is that the outer surface of the skin, this dead skin layer, is heavily colonized by many microorganisms which do not cause disease. These are called the normal flora of the skin. Now, the job of the normal flora is that these are organisms that live in your skin. However, because they've colonized most of the available surface, and because they themselves don't ordinarily cause disease, they are essentially themselves, they form a barrier preventing potentially pathogenic microorganisms from getting a foothold and colonizing that area. And so it's not just the skin itself, it's the normal flora, the microorganisms that colonize this dead layer of skin cells, which don't cause disease. These take up the available surface and are using the available resources in that layer, and thereby prevent potentially damaging microorganisms from getting a foothold in that layer. Now, we mentioned the stomach, so the digestive system itself is continuous with the external surface of the body and also continuous with the external environment, because it's a route through which materials in bulk are taken into the body from the external environment. And so germs can enter the body in food and drink, and most of these are killed by the acid made by the glands in the stomach wall and then digested by the digestive enzymes in the stomach and in other parts of the digestive system. And so, if we were to look at a cross-section of the wall of the stomach, we would find these gastric pits. And at the bottom of these gastric pits, we find these gastric glands, which produce not just the acid, but also the protease enzymes. So the wall of the stomach is lined by columnar epithelium. The columnar epithelium contains mucus glands, which produce the mucus that coats the epithelial layer, but also this mucus doesn't just lubricate the movements of food, and it doesn't just protect the columnar epithelium from the actions of the digestive enzymes and the acid. It also is another barrier that prevents the entry of microorganisms in the food into the tissues of the body via this columnar epithelial layer. So again, we have these gastric glands. These secrete the gastric juice, which contains the hydrochloric acid and also pepsin, a protease enzyme. So the dust and the germs in the atmosphere don't usually get down to the lungs. They're trapped in a stream of sticky mucus made by cells with these tiny finger-like projections called cilia. This layer of cells is called ciliary epithelium, and this covers the nasal cavity, the trachea, and the linings of the bronchial tubes. So the cilia on the surfaces of this ciliated columnar epithelium have a layer of mucus over them, and this mucus traps airborne particles and microorganisms as the air passes from the nasal cavity down into the trachea, the bronchi, the bronchioles, and eventually down into the lungs. So here we have a cross-section of the ciliary epithelium which lines the airways, and here we can see these tiny cilia, and the cilia cause the mucus and trapped particles such as the dust to flow upwards through the respiratory system by this wave-like action. The cilia themselves move in this rhythmic fashion, and this carries the mucus upwards. Eventually, the mucus is swallowed into the esophagus, carried down into the stomach, where the microorganisms trapped in the mucus are destroyed by the stomach acids. So here we see one of the goblet cells where we find in this epithelial layer, it's these goblet cells which produce the mucus. Here are the ciliated cells of the columnar epithelium lining that airway epithelium. And so the flow of mucus is upwards through the respiratory tract, eventually to the back of the throat where the mucus is swallowed down into the stomach. Now, the circulatory system itself has a number of protective mechanisms. So one of these is blood clotting. When we cut or graze the skin, a defense mechanism is triggered, and this is blood clotting. So what happens is that the broken area of the skin eventually starts to form these insoluble fibers across the broken area. Now this happens as a result of the contact between the blood and the atmosphere. This triggers the production of fibrin, which is an insoluble fibrous material which lays itself across the cut in the skin. This then causes the blood cells, the red blood cells and also platelets, to get trapped into this fibrous layer due to the broken capillaries releasing these blood cells and cell fragments. These then get trapped in this fibrous layer. Now another thing that happens is that white blood cells will migrate out through the walls of the capillaries towards this region of damage, and it's these white blood cells which then go on to break down any microorganisms, any pathogens which may have invaded from the external environment into that cut. So it's the blood platelets, the fragments of blood cells, these platelets, which produce these fibers, which are made of this protein fibrin. Now, this is made from a soluble precursor plasma protein. So within your blood plasma, there is a soluble protein called fibrinogen, which normally just flows through the blood and doesn't really do anything. But when you have a cut or a bruise or internal damage, the fibrinogen will form fibrin, and fibrin is insoluble. It forms these fibers which then stick to the edges of the cut and form this meshwork of fibers. Red blood cells then get trapped in these, clog the mesh, and the blood flow is then stopped. This prevents the leakage of blood through that cut, and this is what eventually forms the scab. Now, the function of this blood clot is to prevent dirt and most disease-causing microbes from entering the body through this breach in the external physical defenses. So slowly the mesh and scab contracts, and this pulls together the edges of the cut tissue, and eventually this will help to form new tissue as the scab is broken down, again by the actions of white blood cells. So platelets producing fibrin fibers block the wound. So here we can see the fibrin fibers, and here we can see the platelets which actually form those fibrin fibers which radiate outwards from the platelets. And here we can see the red blood cells trapped in this meshwork, and it's this combination of the fibers and the blood cells trapped in those fibers which will eventually form the blood clot. So here we can see a false color electron micrograph showing the red blood cells trapped in this fibrous network of fibrin produced by the platelets here. So the white blood cells are actually extremely important in both the non-specific and the specific immune response, and it's the white blood cells which form the cellular defense mechanism within the immune system against pathogens which have entered the body. So white blood cells can work in one of three general ways. So firstly, they many of these white blood cells, not all of them, but many of them, have phagocytic activity. They can actually ingest pathogens and digest them and break them down. Other kinds of white blood cells, B cells and plasma cells, can produce antibodies, which are proteins which target particular pathogens to destroy them. And they can also produce antibodies which act as antitoxins to counter the poisons released by certain types of bacterial pathogens, for example. So we can see here that white blood cells are involved in non-specific immunity as well as many aspects of the specific immune response as well. Now, the types of white blood cells which show phagocytic activity, these do so when they encounter any kind of foreign cell, such as a bacterium or a protozoan, and they are not specific. They simply will digest or ingest by phagocytosis, and then digest and break down any kind of foreign or abnormal cell that they encounter. So this is an aspect of non-specific immunity. So phagocytic white cells can pass through capillary walls to engulf and digest, or phagocytose, bacteria in tissue fluid and also in wounds. Now there are two general types of phagocyte. We have monocytes, which form somewhere between 2 and 6% of white blood cells, and these are made in the bone marrow. We also have neutrophils, which make up about 60 to 70% of all white blood cells. These two general types of white blood cells are both phagocytic. They both show this phagocytic activity. So here's a diagram showing phagocytic white blood cells, neutrophils, passing through a capillary wall to destroy bacteria. And so capillaries, again, have walls which are one cell thick. They're made up of a particular kind of epithelium called endothelium, and these are very thin and are made up of a wall of cells only one cell thick. Now, the white blood cells can actually squeeze themselves in the gaps between these capillary endothelial cells in order to get to or pathogenic bacteria in the surrounding tissues and destroy them by phagocytosis, as being shown here. So this is the wall of a capillary. Here we can see the microbes being engulfed and digested by phagocytic white cells, which have migrated by squeezing themselves through the gaps between the capillary endothelium. So we've talked a lot about non-specific immunity. Let's now talk about the different types of specific immunity. So specific immunity is the second but very important line of defense. Now, this occurs once the pathogenic organisms have breached your physical and chemical barriers. And so specific immunity is the second line of defense in the sense that this occurs once the pathogens have gotten inside your body, into your body tissues. And so this happens largely as a result of the bacteria getting through those external defenses, the skin, the eyes, and so on. So specific immunity occurs when the body manufactures antibodies in response to the detection of the presence of invading pathogenic organisms such as bacteria and viruses. Now, your body can detect the presence of these pathogens because they have chemicals called antigens on their surfaces. Now, an antigen is a molecule, a type of protein or other molecule which is foreign to your own body and can be used to identify the presence of foreign organisms such as bacterial cells. So bacterial cells have antigen molecules, for example, in their cell walls, and these antigen molecules can be distinguished from the molecules which your own body cells produce because they are unique in their shape and in their chemical structure. So the slime layer of bacterial cells is often used to hide the antigen molecules of the bacterium from your own immune defenses. So here is a diagram of the H5N1 virus, which causes bird flu, and the virus itself also has unique molecules, antigens, on its surface, in this case protein molecules, which are very different from the protein molecules of your own cells, and as such they can be recognized by the cells of your immune system as being foreign to your body. So specific immunity is a complex, highly evolved biochemical defense system which can not just recognize foreign antigens and mount a response to them, but can also memorize those antigens. In other words, once you've been infected by a particular pathogen, your immune system can remember those antigens from that pathogen so that if you are infected again in the future, the secondary response by your immune system is much more rapid and can kill off that organism much more effectively and much more quickly. And so this specific immunity, the memory of your specific immune system, is the basis of the way that vaccines work. So specific immunity, as we said before, can be active or passive. Now, active immunity can be naturally acquired, And so this happens when you are infected for the first time by some illness, and then your body remembers that particular pathogen by remembering the antigens that it recognized from that pathogen, and so that eventually you are immune to a secondary infection by that pathogen. And so we see here that active immunity, you can acquire active immunity by having been exposed to a pathogen by infection, but also you can acquire active immunity artificially as well through vaccination. So vaccines use this aspect of your immunological memory by presenting you with the antigens, the proteins or other chemicals which are unique to a particular pathogenic organism. These are injected into your body to allow your immune system to recognize them in the event that you're actually infected by that organism in the future. So the body is said to have an immunological memory which allows it to remember exposure to a particular antigen and then produce the appropriate antibody to fight off that particular pathogen. So when you're exposed again, the response is faster and stronger. So this has helped in the evolutionary success of mammals. So antibodies are complex proteins made by specialized white blood cells called lymphocytes. Now, the particular kind of lymphocytes that we're dealing with here are B lymphocytes and also plasma cells. So the antibodies work in 3 different ways to destroy the microorganisms which cause disease. So here we can see a molecular model of an antibody, and this shows you the complexity of some of these molecules. So we have various kinds of lymphocytes which produce antibodies, and each antibody works in 3 different ways to destroy the pathogens that cause disease. So, firstly, lysine antibodies can destroy antigens by engulfing and destroying the antigens themselves. So, if we imagine a simplified diagram of the lysine antibodies, and here we can see an antigen molecule. This is an antigen from a particular pathogen. So remember, the antigen, the word antigen means a unique molecule which is found only on the surface or within a particular pathogen, a particular disease-causing organism. And so the way that these lysine antibodies work is that they have the specific shape, the complementary shape, which allows them to bind to the antigen molecule and then destroy and dissolve that antigen molecule. So this is one mechanism by which particular kinds of antibodies called lysine antibodies can destroy the antigens on certain types of pathogens. And if you imagine these antigen molecules are on the surface of a bacterium, the destruction of these antigen molecules frequently will lead to the destruction of the pathogen, that bacterium. But also remember, antigens can also be the toxins produced by bacteria. And so if you have a bacterium like tetanus, which is producing a toxin, that toxin is frequently a protein molecule which is unique to that bacterium, that microorganism. And so the antigen in that case would be the toxin molecule, and in that case the lysine antibodies would destroy that toxic molecule and ameliorate the symptoms of the disease caused by that toxin. Now, another type of antibodies are called opsonins. So opsonin antibodies stick to the surface of antigen molecules until that microorganism is entirely covered by those antibodies. Now, this appears to enhance the attack from phagocytic white blood cells, which can then go on to engulf and digest those microorganisms. And so opsonins, the way that these antibodies work is that, again, they have the complementary shape which allows them to attach to the antigens, this time on the surfaces of bacteria, for example. And by doing this, first of all, this immobilizes the bacterium. It prevents it from swimming through your body tissues and colonizing them. And it also causes them to be surrounded in such a way that phagocytic white blood cells can more easily identify, phagocytose, and destroy these microorganisms. And so here we can see the opsonin antibodies, and these then stick and bind with the complementary antigens on the surfaces of these bacteria. And so by doing this, we have the bacteria surrounded by opsonine antibodies, and then this allows the phagocytic white blood cells to more easily find them, identify them, phagocytose them, and destroy them. Now, the third mechanism by which antibodies generally work is by agglutination. So agglutinin antibodies will stick to antigens and then cause them to clump together. And so this not only immobilizes these pathogens, it also provides a much larger target for phagocytic white cells to come along and destroy them in large numbers very efficiently. So the agglutinin antibodies are shown here. You can see that they have the complementary shape to bind with antigens on the surfaces of these pathogens in more than one location, and this allows them to clump these together to agglutinate them. This is called agglutination. So we see here agglutinin molecules attached to a particular bacterium on its surface antigens. Now, the agglutinins will then stick them and clump them together, or agglutinate them, and this prevents them from reproducing, it prevents them from moving. It also provides a much larger target so that a single white blood cell can come along and ingest by phagocytosis and destroy large numbers of these bacteria. Okay, so the antibody-antigen reaction is highly specific. Now, this means each type of antibody produced by the immune system attacks only one specific antigen and no others. So the antibody against the measles virus will destroy only the measles virus, for example. So your body needs to produce different antibodies to combat each type of antigen, but once the antibodies have been produced, these then remain in your body, and also the ability to produce those antigens is remembered by the cells of your immune system, and this essentially provides you immunity to that particular pathogen. So antigens are present on many particles, such as the surfaces of bacteria, viruses, fungi, but also inhaled dust and pollen grains. Foreign red blood cells and transplanted tissues also have foreign antigens on them. So it's the antigens present on various forms of food molecules, pollen grains, cat fur, and so on, which can trigger the immune system to respond in an allergic reaction. Now, antitoxins are also part of your body's chemical defense system against disease, and these are a particular type of antibody which will target the antigens, those toxins. Now, many pathogens harm your body by producing and releasing poisonous toxins. We've already seen a couple of examples of this. Now, your body reacts to this by producing an antitoxin which combines with the toxin and renders it harmless. Now, antitoxins are mostly made in the liver. Some can be generic, targeting a wide range of different toxins, but others can be highly specific in their action. Okay, so I see that we're coming up to the first break in this webinar, so I will go for a 5-minute break shortly. But before we do, let's just quickly have a look at immunological memory. So here we see a photograph of an unfortunate boy with whooping cough. So this is caused by exposure to bacterial antigens. Now this affects his lymphocytes, resulting in the production of a particular kind of lymphocyte called memory cells. Now, the large population of memory cells in the immune system then provides protection against the same antigens, and this means that any secondary infection, any infection of by the same organism at some later date, means that it will trigger a much more rapid and much more comprehensive and large-scale production of antibodies. And this means that the second time that you're infected by the same organism, usually the symptoms of the disease are either much less or won't occur at all. So after having been exposed naturally to an antigen, as in the case of having suffered an infection, the person will have developed naturally acquired active immunity. So vaccination or immunization is the process by which an individual has a foreign antigen from some particular pathogen introduced into their body, and the purpose of this is to stimulate a primary immune response, the production of antibodies and memory cells. So the process of introducing the antigen is called inoculation. So vaccination is a form of artificially acquired active immunity. So this is the distinction between active immunity and passive immunity. So active immunity is the actions of your immune system in identifying and then targeting and destroying particular specific antigens. And so passive immunity is received immunity. So another example of passive immunity is when you have the natural transfer of antibodies produced by the mother to the child during pregnancy. So a passive form of specific immunity can result from the natural transfer of antibodies from one organism to another. So the antibodies are not actively made by the child, they are borrowed from the mother in this case, and this happens through the placenta. So naturally acquired passive immunity, this happens in this particular context when antibodies produced by the mother's immune system are transferred across the placenta to the fetus. Also, antibodies can be transferred from the mother to the baby in the colostrum in milk during early breastfeeding. And so this is one of the reasons why breastfeeding is considered to be superior to feeding using formula from bottles, because the breast milk actually contains antibodies which help to protect the baby while the baby's own immune system is developing. So some bacterial infections can cause harm by the release of toxins, as we've said already. Now, certain antibodies can act as antitoxins to neutralize those toxins. But in these kinds of infections like diphtheria, the body may not be able to produce the antitoxin in sufficient amounts in a quick enough time to save that organism. And so a serum containing specific antitoxins can be artificially administered. So the specific serum is derived usually from horses which have been given increasingly higher doses of that toxin. Now, the way that this is done is eventually about 2 liters of blood are removed from the horse to produce the specific antitoxin, which is then administered to the patient. This form of specific immunity is passive, but it's also artificial. Now, unlike maternal transfer of specific immunity, which is natural, which we saw in the previous example. So many people are allergic to the serum made by this artificial process, and unfortunately this can sometimes lead to a fatal immune reaction. Now, penicillin is now used to treat diphtheria, and we're vaccinated against the disease with the DTP-Hib injection, which is administered usually when a baby is only 2 to 4 months old. Okay, so I see that we've come to the next 5-minute break. After the 5-minute break, you will see a poll pop up on your screen, those of you in attendance. If you'd like me to continue with the topic teaching, you can vote for that. If you'd prefer me to go over the exam questions in the order in which they receive votes, I'll happily do that as well. So, okay everyone, we're back after the 5-minute break. I see that a lot of interesting questions have been asked in the Q&A panels for the 5 different Zoom webinar rooms. Keep those questions coming, everyone, and please do take the time to not just ask your own questions, but if you see a question that has been asked by another student, please do upvote that question if you feel like you particularly like that question to be answered. I'm going to be covering those questions in the 30-minute Q&A section towards the end in the order in which they receive votes. And of course, any questions that occur to you after this webinar, you can always go to the private resources page after you log into the website, those of you in attendance, and then you can ask your questions there either in the live chat, or if you prefer to send me your questions privately, you can use the private messaging function there, and I'll continue to be answering questions and providing revision support to the attendees at that page all the way up to the end of your exam period in late June of this year. All right, so it looks like the majority of you have voted for me to continue with the topic teaching, so let's move on with this idea of specific immunity. Now, you need to be able to distinguish between whether or not specific immunity is passive or active. One of the common errors that I see students making in exams on this topic is that they will often confuse the different ways in which immunity is categorized. Either they will confuse specific immunity with non-specific immunity, they might confuse active specific immunity with passive specific immunity, or they might confuse naturally acquired specific immunity with artificially acquired specific immunity. community. So you really have to have a good handle on the definitions of those terms and how to distinguish between them. So let's look at a couple of examples here. So this graph shows the blood antibody level for two individuals, individual A and individual B, shown by the two different coloured graphs, who have both been injected with an antigen to protect them against the disease. So here's a question for you to answer. Explain which person, A or B, received an injection of serum containing antibodies, and which received a proper vaccination containing antigens. So I'm going to pause here and allow those of you in attendance to answer this question. So again, if you don't see the question pop up as a multiple choice on your interface, then you can go ahead and type that into the chat, or type your answer into the chat. So what we have here is two individuals, one of which received a serum containing antibodies against that disease, and one of them received a vaccination containing the antigens from the pathogenic organism. And so we see here the blood antibody level for the two individuals in arbitrary units. This is the amount of antibody per unit volume of blood, and this on the horizontal axis we see the time after immunization in days. So immunization either by serum or by vaccine occurred at time equals 0. Okay, so most of you managed to get the correct answer. Well done. It is in fact person A. So the reason we know this is that person A, we saw that the antibody level rose very, very rapidly. So this tells us that person A received the serum, whereas person B received the vaccine containing the antigens. Now, again, the way that vaccines work is that your body is presented with the antigen or antigens, particular molecules which are unique to a particular microorganism that causes disease. This allows your immune system to recognize those antigens first and then build the complementary antibody molecules. Again, the antibody molecules will fit the antigen because they have the complementary shape. So person A received the serum, person B received the vaccine containing the antigens. Okay, so we've talked about the natural control of disease. In other words, the way in which your body, for example, will respond to infectious illness. And we categorized the defenses of your body as being either the physical and chemical barriers to infection and phagocytosis, which are the non-specific defenses, non-specific immunity. We also talked about the specific immune response, which involves the production of antibodies to the antigens of that pathogen. Let's now talk about the way in which we can artificially protect against, not just protect, but also to treat infectious illnesses. In other words, medical control. Now, various diseases will cause various symptoms, and the medical control of those diseases will treat both the symptoms and also the underlying cause, the etiology of that disease. So some medications, such as painkillers, don't kill the pathogens causing an illness, but they merely relieve the symptoms. We also have antibiotics. Now, antibiotics are medicines which are prescribed for bacterial infections in particular. Antibiotics don't kill viruses, they kill bacteria. So the first antibiotic to be developed was penicillin. Now, Penicillium is a well-known fungal mold which grows on bread and also on common fruits. Its spores are very widespread in the air, and they can easily contaminate culture dishes and also fruits that have been left out for too long. So here we can see Penicillium mold growing on an orange. Now, the Penicillium mold, if we magnify it, we can see the actual hyphae, the microscopic strands which form the mold, but then these can produce fruiting bodies called sporangia, which can then release spores into the atmosphere to allow that mold to colonize other objects. Now, in 1928, Alexander Fleming was investigating the bacterium Staphylococcus and had grown many colonies in culture dishes. Now, after returning from a holiday, he had to throw away many of his culture dishes because they were contaminated by fungal growths. But he also noticed a zone around the fungus where no bacteria had grown. He went on to isolate and extract a chemical from the mold, which he identified as being from the penicillium family, and he named this active agent which caused or killed the bacteria penicillin. So this is a molecular model of penicillin. So he then went on to investigate the action of penicillin on a variety of different bacteria, and he found that it was effective against a large range of bacterial diseases such as scarlet fever, pneumonia, gangrene, diphtheria, syphilis, gonorrhea, and spinal meningitis. Now, mass production of penicillin was then developed in the US just before World War II started. Thousands of injured soldiers and civilians would undoubtedly have died from infections from these types of illnesses, such as gangrene in particular, if it had not been for the mass availability of penicillin. And so the discovery and the mass production of penicillin was incredibly important, not just in general, but in the particular context of the time in which it was discovered. Many of the war wounded in World War II would have died from their wounds as a result of gangrene and other infections, but because of the mass availability of penicillin, these people went on to survive. Now, antibiotics only work against bacteria, and the way in which they work is that they firstly dissolve into the bloodstream. They are then taken in by the bacteria, and the antibiotics are toxic to the bacteria but not to the patient. So some people can have an allergic reaction to some antibiotics, and therefore great care has to be taken in prescribing the correct antibiotic to the right person. So antibiotics generally will work in one of two ways. Either they will interfere with the structure of the bacterial cell wall and disrupt the structure of the cell wall, and an example of this is penicillin, or they can interfere with the internal biochemical reactions of the bacterium itself. In other words, they interfere with the metabolism of the bacterial cell. Now, an example of an antibiotic that does this is streptomycin, which is another commonly prescribed antibiotic. Now, viral infections such as colds, flu, herpes, and AIDS, measles, and so on, these cannot be treated with antibiotics. So viruses don't have cell walls, and they have no internal metabolism of their own, and therefore the two main ways in which antibiotics work on bacteria are not valid for viruses. So it's very difficult to produce medications to treat viral infections because the viral genetic material itself ends up inside your own body cells. Cold sores are a good example. The viral DNA will stay in the body cells of the person for the rest of the person's life and can be activated again by various factors such as sunlight, stress, and so on. Now, only surgical removal of the infected area would remove the virus from the body, and this is not an option for most people. Now, I mentioned antibiotic resistance a little bit earlier on. So, if we imagine a bacterial population, now, bacteria don't just multiply rapidly, they tend to show a very high rate of mutation. And so if we imagine a spontaneous mutation occurring in this growing population, which makes one individual bacterium resistant to a particular antibiotic, if we then imagine this population of bacteria are then subjected to that particular antibiotic, it means that the susceptible individuals, the individuals in that population without that mutation, will then be killed off by that antibiotic. This then leaves the resistant individuals to multiply rapidly and form a completely resistant population of antibiotic resistant bacteria. And so this is a huge problem in hospitals because the bacterial populations in hospitals are being subjected to many different antibiotics. And so the antibiotics are acting as a selective mechanism which kill off the susceptible bacteria and only leave behind the resistant bacteria. So the prevention of antibiotic resistance in pathogenic bacteria is now a huge priority for maintaining public health. To reduce the possibility of antibiotic-resistant strains of bacteria developing, there are a number of strategies that have been adopted in medical contexts. So firstly, fewer antibiotics should be prescribed by doctors. Unfortunately, one of the problems in medicine is that antibiotics tend to be overprescribed. In other words, they tend to be prescribed by doctors not just for bacterial illnesses, but sometimes they can be prescribed in error to patients who have viral illnesses. And so if a person presents with a cold, a flu, or a sore throat, really what the doctor should be doing is either prescribing an antiviral medication or some other medication, but not an antibiotic. So another strategy here is to use antibiotics only for limited periods of time. And so a specific antibiotic should be used for up to 3 years, and then an alternative need to be used. So there are many synthetic forms of penicillin which are prescribed nationally for a set period of time, and then nationally the prescription is then changed to a different antibiotic to prevent that antibiotic leading to the development of resistance in populations of bacteria. So antibiotics should not be made available over the counter or over the internet. They should be only available via prescription. And again, this then limits the use of that antibiotic and thereby reduces the probability of the emergence of some antibiotic-resistant strains. So in the US, most drugs can be purchased over the counter or over the internet, and there's only a few states in the US which don't allow this, and obviously this can be a problem. Now, another way in which we can manage the emergence of antibiotic-resistant strains of bacteria is to limit or rather to harmonize the way in which antibiotics are available across the world. Now, in some countries, antibiotics are not prescription only and can be purchased over the counter, such as in the US. Now, this is obviously a problem. So these antibiotics, amoxicillin, can be purchased by people on holiday, whereas if they go to their home country, they can't purchase them without a prescription. So if we imagine that a person with a persistent cough and catarrh went abroad, that person could then purchase this amoxicillin without a prescription. Now, the person's GP in England would not prescribe these antibiotics, and so this is a problem. Now, it's not just in humans where we need to limit the use of antibiotics. So antibiotics should not be given to livestock and poultry as a matter of routine. Now, unfortunately, this is the case because poultry and cattle farmers, they also want to limit the spread of infection and also treat infected animals. Now, this has been done in the past, and this has led to the spread of antibiotic-resistant strains in cattle. Now, this is a problem for humans because those same organisms will persist in the dairy products, such as the milk, and also in the meat of these organisms when that meat and dairy products goes on sale. So the use of antibiotics to promote growth has recently been effectively banned in the European Union, but not their use as preventative measures or prophylactics. Now, this is also a problem because some farmers will give antibiotics to their cattle even if those cattle are not yet infected. And the idea behind that is to prevent infection by ensuring that these cattle have those antibiotics flowing around in their circulatory systems, so that if they are infected, the infectious organisms are destroyed. Unfortunately, this contributes to the development of antibiotic resistance. So penicillin is produced commercially on a large scale by culturing the mold Penicillium inside large fermenters such as these. The UK has some of the largest pharmaceutical companies in the world, and the manufacture of penicillin is done on a very, very large scale. So a culture of the mold is placed in the fermenter, and the conditions inside the fermenter are then carefully controlled using temperature and other sensory probes. The nutrient mixture inside the fermenter contains sugar and other nutrients such as nitrogen in the form of ammonia and also vitamins, all of which the Penicillium mold needs to multiply. The pH is maintained at about 6.5 and the temperature is maintained at about 30 degrees centigrade. After about 30 hours, the penicillium starts to reproduce and starts to manufacture penicillin in large quantities. So the penicillium has to complete its main growth phase, which is an exponential phase of growth, before it starts to release penicillin into the surrounding nutrient medium. The release of penicillin takes about a week to complete. After that, the mixture is drained out of the fermenter by opening the tap at the bottom, and it's collected into another vessel. The mixture is then filtered and purified, and the penicillin is then extracted in a salt-like form. At the end of the batch process, the fermenter is completely emptied and then sterilized again, ready for the production of another batch of penicillin. So, another one of these topics that many of you voted for me to cover was vaccination. Now, we've already mentioned this in principle, the way in which it works. The human body naturally has the ability to use active immunity to prevent and cure infection. Vaccination is basically a means by which we can exploit this ability to acquire active immunity artificially. So a vaccine is a preparation of a disease-causing agent, its components, or its products, and the idea of the vaccine is to induce or to cause active immunity in the recipient of the vaccine. Now, modern medicine relies heavily upon the exploitation and simulation of natural processes of active immunity. The production of vaccines containing antigens which stimulate the adaptive immune response was first pioneered by an English country doctor named Edward Jenner in 1756. So, in the late 18th century, smallpox was a much feared disease, and a smallpox epidemic hit Edward Jenner's home county of Gloucestershire in 1788. Now, at this point, he made a number of important observations about the spread of the disease. So firstly, he observed that milkmaids and other people who came into contact with cattle rarely suffered from smallpox. He also noticed that these same workers did come into contact with a disease related to smallpox but much less virulent, called cowpox. So he theorized that the pus in the blisters which the milkmaids received from cowpox protected them in some way from the more virulent disease, smallpox. In May 1796, he tested this theory, or this hypothesis, by inoculating the young James Phipps with a material extracted from the cowpox sores found on the hand of a milkmaid. So, Phipps became ill with cowpox, and several weeks later, Jenner injected him with material containing the smallpox virus. Jenner actually also vaccinated his own son, and this is shown in this statue. James Phipps survived and did not suffer from smallpox. So, Jenner made the conclusion that infection with cowpox provided immunity to smallpox, and that laid the foundations of the practice of vaccination. Now, we now know that this happened because cowpox and smallpox viruses have a similar antigen, and therefore the acquired immunity worked against both diseases. At first, Jenner's work was met with skepticism and also some ridicule. However, following further work and technique improvement, Jenner's discovery was eventually accepted, and in about 1840, a smallpox vaccination was provided to the British population free of charge by the government. So, when you're given a vaccine, your body reacts as if it were suffering from the full infection, even though the vaccine itself does not contain usually the live pathogen, but the antigens, the proteins and other substances from that particular pathogen. The white blood cells start to produce antibodies to destroy the pathogen and prevent further infection or replication of that pathogen, in this case the virus. Now, the antibodies can then stay in the body for some time, and certain cells in the immune system remember that antigen and can produce the correct antibody at short notice. This gives rise to the long-term immunity that you have as a result of that vaccination. Today, vaccines are widely used to protect children and adults from a variety of different types of infectious diseases. So polio vaccine is being administered here orally as a liquid. Some vaccines don't need to be injected, they can be orally administered. Other vaccines need to be injected, so the MMR vaccine, which protects children against measles, mumps and rubella, is administered as an injection. Now, vaccines are given at set intervals during the first 5 years of a child's life, and they are stored typically at low temperatures to prolong their effectiveness and also to have them ready in large quantities in the event of an outbreak or an epidemic. So another one of these topics that many of you voted for on the private resources page prior to this webinar was monoclonal antibodies. So let's take a look at those. So antibodies are produced by the B lymphocytes of the immune system in response to foreign antigens. Antigens are either proteins, glycoproteins, or polysaccharides, and these can be either free molecules, or they can be located in the surface of an invading microorganism. They can also be present on the cell membranes of transplanted organs that you receive from another human individual. Now, antigens are present on all cells in an organism, but normally will only provoke an immune response if they are foreign to that organism. So, B lymphocytes differentiate from stem cells in the bone marrow and then migrate to specific regions of lymphatic tissues. And each lymphocyte has about 100,000 surface receptors which are specific for a foreign antigen, for a particular antigen. So here we can see a simplified view of a B lymphocyte, and here we can see a B cell antigen receptor. Now, a vast array of B lymphocytes bearing different cell surface receptors are found in the lymphatic tissues, in the spleen, in the lymphoid, in the lymph nodes, and so on. So when a specific B cell or B lymphocyte encounters a specific non-self or foreign antigen, and this triggers a phase of what's called clonal expansion. Clonal expansion is a natural process within the immune system where the cell that encounters that antigen multiplies itself rapidly. So when B cells are activated through the binding of a particular antigen to their complementary cell surface antibody receptor, these then divide to produce a clone of specific B cells, each of which has the cell surface receptor which is complementary to that specific antigen. Many of these cloned cells then differentiate into plasma cells, and these plasma cells are like factories which manufacture large quantities of the antibody to that particular antigen. The antibodies then circulate in the blood and the lymph tissue, in the lymph fluid, where they then will bind to the foreign antigen wherever that antigen is found, typically at sites where the pathogen which bears that antigen is multiplying and invading the host tissues. So some of the activated B cells do not differentiate into plasma cells, they remain as memory cells in the lymphatic system. So it's these memory cells which persist after that infection has been cleared away and can then go on to more rapidly differentiate to produce clones of plasma cells and produce large quantities of antibodies much more rapidly the second time you are infected by that same pathogen. So, to summarize, a vast array of different lymphocytes with different surface receptors exist in your immune system. When a particular antigen binds to one of those specific B cells, that B cell will multiply to produce a clone of B cells, many of which will differentiate to become specialized plasma cells, all of which will secrete that particular antibody. The antibodies then bind to the antigens at the invasion sites and rapidly facilitate the destruction of that pathogen. But many of those, that clone of B cells will persist as memory B cells, so that the second time you're infected by that same microorganism, the response is much more rapid. So antibodies clearly have a great potential for use as therapeutic and diagnostic and research agents, but for this, substantial amounts of a single specific antibodies and a particular antibody are required. And so in 1975, two scientists, Kohler and Milstein, pioneered a method for culturing cells that allowed them to harvest large quantities of pure, highly specific antibodies. These were called monoclonal antibodies. Now, they successfully grew a population of B lymphocytes derived from a single ancestral B cell. This population of B lymphocytes allowed for the harvesting of a single kind of antibody, so these were referred to as monoclonal antibodies. Kohler and Milstein had to solve a problem. Normal antibody-secreting plasma cells don't survive and divide outside of the body. The solution was to hybridize the antibody-producing cells with human tumor cells which do grow outside the body. The resulting cells were referred to as hybridoma cells, and as they're all clones of a single line of cells, they can be used to harvest specific antibodies. So, monoclonal antibodies have great potential advantages over conventional treatments for all types of cancer and other tumours. Conventionally, the circulatory system has been used to carry cancer drugs all around the body in a non-specific manner. So the drug enters both the healthy and the cancerous cells, often causing serious side effects to healthy cells, as well as killing the cancer cells. Similarly, radiotherapy also affects healthy tissues in the region of the tumour, and this can also cause unwanted side effects. Therapeutic drugs can now be attached to monoclonal antibodies, which will deliver the drug to the antigens on the surface of specific cancer cells that are to be treated, and thereby avoid harm to healthy cells. So, for example, Herceptin is a monoclonal antibody which can bind to a particular type of protein called HER2, which is only found on the membranes of certain types of breast cancer cells. So in this way, we can attach a anti-cancer medicine to this Herceptin antibody, which can then specifically target that particular anti-cancer drug to only the tumour cells, and thereby avoid the typical effects of chemotherapy on healthy cells. Now, radio-labelled monoclonal antibodies which are labelled with radioactive isotopes, are being increasingly used to target the location of tumours and blood clots so that they can be more effectively imaged on radiographs. So in the case of deep vein thrombosis, which is a type of blood clotting which occurs inside the deep veins of an individual, we can use these radio-labelled monoclonal antibodies to target fibrin. So in this case of DVT, the monoclonal antibody used is radioactive antifibrin, which combines with the fibrin at the sites of the clot. So DVT is a rather serious condition which affects for example, surgical patients and people who are immobile for long periods of time, and this affects 2.5 million people every year. The ability to radioactively label these monoclonal antibodies, which can then bind with the fibrin at the sites of these blood clots, allows us to more accurately identify where these blood clots are happening and then treat them accordingly. So we've been talking a lot about the immune response in animals like ourselves, but plants also have specific defenses against the invasion by infectious microorganisms. Now, there are other forms of plant diseases which are non-infectious, which result from the lack of certain minerals. So we need to kind of cover both the non-communicable diseases in plants, which result from the deficiency of certain minerals, but we also need to look at the defense responses of plants to infectious organisms such as viruses. So let's start by looking at the kinds of minerals that plants need, and these are the types of minerals which are present in plant fertilizers. So we have potassium nitrate, which contains potassium and nitrogen. We have ammonium sulfate, which is another source of nitrogen for the plant. We have ammonium nitrate, which is yet another source of nitrogen. We have ammonium phosphate, which is a source of both phosphorus and nitrogen. And we have urea, which is another source of nitrogen. Now, phosphorus and nitrogen, as well as potassium, magnesium, and other minerals, are used by plants for various purposes. So, 4 mineral ions are essential for healthy plant growth. These are nitrogen, potassium, phosphorus, and magnesium. Lack of one or more of these specific minerals can result in the plant showing deficiency symptoms. Now, nitrogen is taken in by plants usually as nitrate ions, phosphorus as various kinds of phosphate ions, potassium and magnesium are found as K+ and Mg2+, but these are all required for the metabolism of a healthy plant. So nitrogen is a major element needed by plants. It's absorbed from the soil as nitrate ions. These are essential for producing amino acids, which are then used to form proteins. Now, unlike heterotrophs like ourselves, plants generally don't consume proteins and then digest them down to make amino acids. They need to construct those amino acids from scratch, and for this they need nitrogen. Now, if they have a nitrate deficiency, this tends to manifest itself as the upper leaves remaining green and healthy looking, whereas the lower leaves start to become yellow and discolored. So lack of nitrate ions can result in stunted growth, and nitrate is often referred to by farmers as the leaf maker because a lack of nitrate results in the paling of upper leaves and yellowed lower leaves. So closer examination of the upper leaves of a plant with nitrate deficiency reveals that yes, they are green, but they do look a little bit paler than they would in a healthy plant. But typically the symptom of nitrate deficiency, which is spotted first, is the yellowing of the lower leaves and the dying off of the lower leaves. So the only group of plants that don't need to take nitrogens directly from the soil are leguminous plants such as beans, peas and clover. Now the reason for this is that they actually contain colonies of microorganisms, colonies of bacteria in their root nodules. So this is a photograph of the roots of a leguminous plant. These are the root nodules. Now the root nodules contain a type of bacterium called nitrogen-fixing bacteria. So these can fix the nitrogen into nitrates and then make them directly available to the plant. And so these bacteria live in a symbiotic relationship with the plant, where the plant provides them with shelter from the external conditions, while the bacteria provides the plant with a very important nutrient in the form of nitrates. So phosphorus is also an important element required for healthy growth in plants. It's absorbed from the soil as phosphate ions dissolved in the soil water. Now phosphate is important for a number of reasons, partly because it's a essential part of the energy storage molecule ATP, but also because phosphate is essential for producing DNA, for dividing cells, and so on. Now, phosphorus deficiency usually manifests itself as not just stunted growth, but small purple leaves and poor root growth. So here we can see some maize seedlings. So this row are the healthy maize seedlings, which have received the correct amount of phosphate nutrients. Here we can see the stunted growth of that occurs when the same plants receive lower amounts of phosphate. So potassium is another important element required by plants. Potassium again is dissolved as potassium ions in the soil water, and these are essential for controlling the ability of the plant to take up water and reduce its water loss by closing the stomata. So potassium deficiency usually manifests itself as the yellowing of patches of leaves with dead spots in those leaves, as being shown here. So the lack of potassium ions results in not just an unhealthy yellow appearance to the leaves, but also these very obvious dead spots on the leaves. So magnesium is another one of these mineral ions required for healthy growth. Now in this case, magnesium is needed for the production of chlorophyll. It's absorbed again from the soil as magnesium ions dissolved in the soil water, and so magnesium deficient soils will result in leaves having normal looking upper leaves, but yellowed and kind of mixture of yellow and green lower leaves. Now the yellowing is due to the lack of production of the green pigment chlorophyll. So again, we see this yellowing towards the lower ends of the plant, but in this case we also see a kind of progressive decrease in the greenness of the leaves as you go from the top to the bottom of the plant. Now, the earliest terrestrial land-dwelling plants evolved from aquatic ancestors that thrived in a period of time over 450 million years ago called the Ordovician period. So these simple plants were very different from the highly evolved terrestrial plants of today. They lacked the vascular tissue, the xylem and the phloem, that allows water and other nutrients to be transported through the plant. Now, the true vascular plants which contain these transport tissues started to appear about 400 million years ago and continued to diversify throughout the Devonian period. Now, vascular plants developed waxy cuticles and protective coverings to reduce water loss from their tissues, and they also evolved a vascular transport system to deliver water and nutrients to all cells of the plant. They were less reliant on water than their predecessors and therefore more able to survive drought and desiccation. Now, the diversification of flowering plants into the vast array with which we're familiar today started in the Cretaceous period and was coupled with the sudden burst of speciation in insects. Insects are the most numerous herbivores that feed directly on plants, and this accounts for about 30% of all insect species. So there has been a close relationship in the evolution of terrestrial plants and also that of insects. The diversification of insects was a selection pressure for plant evolution. It ultimately led to the selection of plants that had defensive adaptations against insects. So herbivores, also known as primary consumers, are animals that feed directly on plants. Plants are also called producers because they carry out the process of photosynthesis and thereby produce a source of carbon and energy in the form of starch, a carbohydrate which the animals feed on when they feed on the plants. And so plants have had to develop various mechanisms to protect themselves against the feeding behaviors of herbivores, of primary consumers. So let's look at some of those now. Okay, so the first insects to inhabit the earth ate plant material by biting bits off and then chewing the material. So these first insects evolved mouthparts, which allowed them to effectively bite off leaves and smaller stems. Grasshoppers and locusts are good examples of these kinds of insects. So here we can see a diagram of a locust head, and we can see the locust mouthparts are adapted for eating plant material such as grasses and related cereal crops. Now, as vascular plants continued to evolve, later herbivorous insects developed more specialized modes of feeding. Some evolved piercing mouthparts that allowed them to penetrate the stems of the plant to feed on the sugary materials found in the phloem tissue. Aphids are examples of these types of feeders. So other insects developed mouthparts that allowed them to reach deep inside flowers, where the nectaries are usually situated. Now, the nectaries are the organs of the flowers which produce the sugary material nectar. So the nectar is an energy-rich food and represents a good source of energy for these kinds of insects. So here we can see the head and proboscis of the butterfly. Here we can see that the proboscis is coiled, but when this organism feeds on the flower, it uncoils the proboscis to allow the end of the proboscis to reach the nectary deep inside the flower. Within the proboscis, you have muscles which contract to cause the movement of food through the cavity in the middle of the proboscis. So over time, plants have evolved numerous strategies to protect themselves from herbivore attack by insects and other animals and thereby increase their chances of survival and reproduction. Many plants produce secondary metabolites called allelochemicals or anti-herbivory compounds, which influence the metabolism, the growth, or the survival of the attacking herbivorous animal. Now, these secondary metabolites are not involved in the plant's own photosynthetic or metabolic processes, and they're not needed by the plant itself for growth or reproduction. They're produced as byproducts during the synthesis of primary metabolites, which are essential for the normal functioning of the plant. So let's take a look at some of these chemicals. So plants that do not have a defense against against a particular herbivore eating them, face overgrazing or insect infestation, as being shown here. Now, allelochemicals can act in a number of different ways to reduce the impact of that herbivore. So these include the fact that these allelochemicals are toxins which cause either temporary or permanent harm to the organism feeding on the plant. They can also be repellents, which cause the herbivore to move away from the plant and therefore leave the plant material intact. They can also act as agents which reduce the palatability of the plant tissue, and so the taste of the plant tissue itself is caused to be unattractive to the herbivore, and so the herbivore then doesn't continue to eat the plant material. So there are various types of these allelochemicals. And as such, they can be classified into three main groups. They are either nitrogen compounds, such as alkaloids, they can be terpenoids, phenolics. So we've got these three different main types of allelochemicals. Now, alkaloids are nitrogen-containing compounds which are derived from amino acids. They tend to impart a bitter taste to the human palate, and this bitter taste gives the plant tissue that characteristic bitter taste. So these include nicotine, caffeine, morphine, strychnine, and quinine. These are all alkaloids. Now, terpenoids are a large family of organic chemicals derived from the 5-carbon monomer isoprene. Now, isoprene is the basis of natural rubber, and the terpenoids usually have typically aromatic properties. Phenolics consist of an aromatic ring that looks like benzene, and this is joined to a hydroxyl group. So, when ingested by a herbivorous animal, these animals ingest the alkaloids, and the alkaloids can have a variety of effects on these animals. So they can either inhibit or activate certain enzymes in the animal, and this alters the animal's metabolism. They can change the way in which carbohydrates and fats are stored in the body. They can inhibit protein synthesis, which occurs on the ribosomes of cells, and this inhibits the process of growth and repair. They can also interfere with the repair mechanism, which repairs damage to DNA, and this occurs in the healthy nuclei of animal cells. When this is interfered with, this then interferes with the ability of those animal cells to repair the DNA, and this can then lead to cancer in those animal cells. They can also cause the weakening of cell membranes, causing the contents of those cell membranes to leak out, causing the death of those cells. Finally, they can interfere with the transmission of nerve impulses around the animal's body, and so these alkaloids can also be neurotoxins. So, anabasine is naturally present in the tree tobacco plant, and this is a close relative of the common tobacco plant. Chemically, it's similar to nicotine, and it's been used as a commercial insecticide. So we can see here the similarity between the anabasine molecule and the nicotine molecule. Now, atropine is another one of these naturally present alkaloids, and this is found in the deadly nightshade, and also some other related plants. So atropine has a stimulant effect on the central nervous system and the heart, but can be lethal in sufficient concentrations. And so this also acts as a defense mechanism for these plants to prevent herbivores from eating them. Terpenoids, again, are a large family of organic chemicals derived from the 5-carbon monomer isoprene. And this is found in eucalyptus, cinnamon, ginger, menthol, camphor, and also cannabinoids found in the cannabis plant. Now, terpenoids can make either the leaves poisonous, they can lyse or break down red blood cells of herbivores, and so these are various effects of terpenoids, and terpenoids are a very large family of compounds. So here is an example of a terpenoid. You can see here that the molecular structures of these terpenoids can be extremely complex. This one wards off insects and protects against microbes. Now, the third family of plant defense allelochemicals are the phenolics. So these consist of an aromatic ring joined to a hydroxyl group, and these can have the effects of inhibiting digestion by interfering with protein absorption and the action of digestive enzymes. They can also cause the grinding down of an insect's mandibles, the structures that an insect uses for feeding, and so eventually that insect can no longer feed. So here is gallic acid, which is one type of phenolic. Here we can see the benzene ring, which is characteristic of these chemicals. Here is gentisic acid, which is another phenolic. Again, we see that they have in common a aromatic or benzene type ring connected to one or more hydroxyl groups. Capsaicin is a naturally active component of chili peppers. This is another phenolic, and this is characteristic to plants belonging to the genus Capsicum. It's a very strong irritant, as many of you will know from having eaten chili peppers, and this produces a burning sensation in any tissue with which it comes into contact. So capsaicin probably evolved as a deterrent against herbivores. It has no effect on birds, which actually do disperse the seeds of these types of plants. So here is the molecular structure of capsaicin. So pyrethrin is naturally present in the seed cases of the chrysanthemum plants, and extracts of this plant now provide the most widely used range of insecticides in the world. This is a repellent. It repels insects from the plants and kills them if it's present in sufficient concentrations. Now, the way that this works is that it attacks the nervous system of insects. Now, many of you have unfortunately come into contact with the stinging nettles. So the stems and the leaves of the stinging nettle plants are covered with tiny pointed stinging hairs, as you can see here in this magnified photograph. If an animal touches them, they break off and penetrate the skin, and this releases a complex mixture of chemicals which causes a stinging, irritating rash. These stings are designed again as defense mechanisms, and in this case against browsing herbivores such as cattle and rabbits. Some other plants, such as the white dead nettle, mimic the appearance of stinging nettles without actually possessing stinging nettles of their own. So, some geranium species produce amino acids called cuscilic acids, which cause paralysis of various types of beetles. Now, in this case, the Japanese beetle is a significant agricultural pest of these types of geraniums. Now, within 30 minutes of ingestion, the chemical paralyzes the animal. Now, the effects of the chemical only last a few hours, but during that time, the beetle is often consumed by its own predators. So quiscalic acid is thought to mimic L-glutamic acid, which is involved in transferring nerve impulses from nerves to muscles in the insects. Now, there are a number of other mechanisms by which plants defend themselves against herbivores. So celery plants produce a chemical called jasmonate, which they produce when they're attacked by herbivorous insects called earworms. So here is the molecular structure of jasmonate. So the presence of this chemical acts as a signal which switches on the plant's resistance genes that code for insecticides. The celery plant only starts to produce its insecticides when attacked by insects, and it does this to conserve energy. So here we can see an earworm eating a cotton plant. Now, earworms are actually larvae of the moth, Helicoverpa zea, and these eventually become resistant to the insecticide as they switch on another gene in their gut lining that codes for an enzyme to break the insecticide down and thereby render it harmless and ineffective to the insect. Now, bracken displays allelopathy, and this is to say that it produces allelopathic chemicals or allelochemicals, and this is an important factor in its ability to dominate other forms of vegetation, particularly in areas after a fire has taken place. Now, herb and tree seedling growth may be inhibited even after the bracken fern is removed, apparently because the soil, the active plant toxins from the plant remain in the soil. But brackens of the northern hemisphere are used as food plants by up to 40 invertebrate species, including the larvae of some butterflies. Now, the numbers feeding on the bracken increase as the season progresses due to the decreasing level of the toxin and the production of nectaries in the spring. These nectaries provide food for ants, which in turn may kill any herbivorous insects in the vicinity. So the toxin in bracken fern is thiaminase, and this is an enzyme that destroys thiamine, vitamin B1. A grazing horse would essentially suffer from a vitamin deficiency of thiamine, which causes myelin degeneration of the peripheral nerves. Now, the myelin sheaths are the fatty sheaths which surround the axons of peripheral nerves. So a loss of this fatty insulation layer causes various kinds of neurodegenerative illness in the horses. Horses will only eat bracken if no other forage is available. Horses have to eat bracken regularly over 1 to 2 months before developing clinical signs of these illnesses. So the first sign of poisoning in horses is weight loss. Later, weakness and abnormalities with the way that they walk, their gait, are present, and this progresses to staggering, and this is sometimes called the bracken staggers. Affected horses may stand with their legs widely placed and their back arched, and this tells observers that these horses are suffering from bracken poisoning. So the symptoms include muscle tremors, general weakness, and a slow heart rate, or sometimes unusual heart rhythms. Near the end of the course of this disease, heart rate and temperature rise, and the animals can't get up and may have spasms and an upward arching of the head and the neck. The syndrome runs its course, and death usually occurs within 2 to 10 days of the onset of the signs. Okay, everyone, I see that we've come to the second break at the end of the second hour of this webinar. So once again, I'm going to break for 5 minutes. Again, after the break or during the course of the break, you should see on your screens, those of you in attendance, a poll pop up, which will allow you to vote as to whether you'd like me to continue for an extra 30 minutes or to go directly into the Q&A section. I see so far that the vast majority of the votes for the questions that have been posed so far in the Q&A panels have been to do with the development of drugs and how this is done. So again, keep the questions coming. We'll go for a break in 5 minutes, and either we'll continue for an extra 30 minutes before entering the Q&A segment, or we'll go straight into the Q&A section. So I'll see you in 5 minutes' time. Okay, everyone, we're back after the 5-minute break. Let me just have a look at the results of that poll here. All right, so it seems like the vast majority of you have voted for me to go straight into the Q&A segment. That's fine. And it seems like across all 5 webinar rooms, by far and away, the majority of the questions are generally to do with drug design and development. So let's address that. And again, any other questions, any questions that you either didn't ask or occur to you afterwards, or any questions we don't get time to cover, please do go to the private resources page after you log into the website, and you can continue to ask your questions there, and I will respond there. Either you can ask them privately, or you can ask them as a kind of and get a discussion going in the live chat. All right, so let's quickly go on to drug design and development. Let me just find the relevant slide of my presentation here. And again, I will make all of the images of these slides available to all of you on that private resources page as well shortly. All right, so let's look at drug development now. So the word drug in medicine is used to describe any substance which, when taken into the body, will affect the way in which the body systems work. So scientists are constantly looking for new drugs, various different kinds of drugs. And so an example of these relatively recent development are the statins, and these are potentially very important for the control of cholesterol, and cholesterol being a risk factor for many diseases, including many cardiovascular illnesses. So when new medical drugs are devised, they have to be extensively tested and trialed before being used. Now, new drugs can't be licensed for use until all the medical trials are completed. So for the vast majority of you, you are going to need to have an understanding of the main stages in drug development, including clinical trials. So drug development follows a process with 3 distinct stages. The first stage is the so-called preclinical stage. This includes discovery, animal testing, and patent application. So the second stage is the clinical stage. This includes human testing and final marketing. Now the clinical stage is divided up into phases. The first phase is small-scale trials on healthy individuals. The second phase is medium-scale trials on sufferers of the condition for which the drug is intended to treat. The third phase is large-scale trials on the sufferers of the condition. The fourth phase is that the drug is licensed and then prescribed for general use. Now, the third stage in drug development is called the post-market stage. This includes ongoing feedback from doctors, and also the patent may expire and generic drugs can then be produced. So let's go through these stages one by one, because many of the questions that you've been asking me that have received the most votes in the Q&A panels have to do with one or more of these stages. So let's start with the preclinical stage. So obviously the first stage in any development of any drug is its discovery. So historically some drugs that we've already mentioned, such as penicillin, were discovered by accident. Now typically antibiotics, because they are extremely complex in terms of their molecular structure and as such are very difficult to synthesize, typically antibiotics continue to be a major class of drugs which continue to be discovered by accident. But some others, such as quinine, were discovered by the analysis of traditional medicines that were used before modern allopathic medicine came into wide use. So today the development of new drugs is very much a scientific process, part of which is called rational drug design. So clinical, the word clinical means medical treatment for humans. So therefore the phrase preclinical means before treating humans. Rational drug design involves the design and synthesis of compounds based upon the known structure of either a specific target or one similar in structure and function. So researchers in the pharmaceutical industry can use powerful computers to search through databases containing the structures of thousands of different chemical compounds. So here we have a monoclonal antibody selected by a computer as a suitable starting point for a preclinical drug trial. So the computer selects compounds that are most likely to interact with the specific target or receptor, and these are then tested in the laboratory. So preclinical testing is a period which lasts about 5 years typically, and it's during this preclinical testing stage that animal testing takes place. So in this photo, the traditional white lab rat can be seen in the cages. The rats are bred to be white in colour so that if they escaped or were released by activists, they would not survive for too long. Now, the law in Britain requires that all new drugs are tested on at least 2 different species of live mammals, one of which must be a large non-rodent, such as a dog. So we also have testing on primates as well. Now, about halfway through the preclinical stage, the drug company would apply for a patent if the drug had shown real potential. The results of preclinical testing can also be used to determine how the drug should be formulated for its intended clinical use, so for example either as a pill or as an injection. Now, the clinical phase is when trials of the new drug are conducted on human volunteers. So clinical trials, again, are divided into 4 phases. Now, each of those phases seeks to answer different questions about the new treatment. So phase 1, again, is small trials on healthy individuals. Phase 2 is medium-scale trials on sufferers of the condition. Phase 3 is large trials on sufferers of the condition. And finally, phase 4, the drug is then licensed and prescribed for general use based upon the outcomes of the first 3 phases. So all of the trials in phase 1, 2 and 3 are usually randomized double-blind studies involving the use of a placebo to ensure the objectivity of those phases. So we need to be clear about the meanings of these terms. So randomize means to arrange or carry out in random order. Placebo means an inert medication given to the control group during drug testing. So this is often only water or a starch or sugar tablet. Double-blind means a test procedure in which the identity of those receiving the correct medication is concealed both from the doctors and the other patients. So let's have a look at the first phase. Now, these small trials involve only healthy individuals. Now, the purpose of this is to evaluate the safety of the new medication, establishing, for instance, a safe dose range for the drug. During this stage, any side effects of the drug are also identified, and this phase usually lasts relatively short. It usually lasts a few months. Now, the larger scale trials on sufferers of the condition in phase 2, these are designed to test the effectiveness of the new medication on the actual illness for which it's intended. So for this reason, volunteers affected with the relative medical condition are chosen rather than healthy individuals. Now, phase 2 of the clinical stage is longer. It usually takes up to 2 years. Now, phase 3 involves large-scale trials on large numbers of affected individuals that have that actual disease condition. So this typically involves hundreds or even thousands of test subjects. Now, it's during this phase that much more information can be collected about the effectiveness and the safety of the drug. Now, phase 3 can take several years, and once the trials are completed, the treatment can then be licensed for general use if it's found to be both effective enough against the desired condition and also safe enough. So again, phase 3 is really the kind of make or break phase for drug development. So now, phase 4 is when the drug is licensed and prescribed for general use. In other words, it becomes available for the pharmacist, and usually only by prescription at this point. Now, as we saw before, many drugs or almost all drugs, are produced at a large scale in industrial manufacturing facilities. So as I said before, the UK actually has some of the largest pharmaceutical companies in the world, and as we can see here with these fermenters being used to manufacture insulin, many of these drugs are manufactured on the industrial scale, either by natural processes such as fermentation or by chemical synthesis. Now, once the drug has been prescribed and is available for general use, it enters the post-market phase. Now, it's during this phase where feedback from GPs on the effectiveness or otherwise of the new drug occurs, and also patients' feedback on the effectiveness and the safety of this drug. So it's during this phase where further evaluation is done on the safety and the effectiveness of the drug in the long term. So during the post-marketing period, eventually the patent on that drug will expire, and then other drug companies can make their own generic version of the drug. The generic drugs typically will cost much less than the branded variety, and the 20-year patent allows the drug company to recover the costs that they outlaid on research and development, and also to make a sufficient profit. After a long enough period of feedback from the medical profession, the drug may no longer be prescription only and may become available over the counter. Okay, everyone, so that hopefully answers the majority of the questions on the subject of drug development. Let me now have a look at some of the other questions that were asked. Okay, so it seems like the questions which have received the second highest number of votes across all 5 of the student groups are to do with drug side effects and what happens when the side effect of a drug is found to be particularly serious. All right, so let's have a look at this in a little bit more detail. So let me give you a case study which some of you may have come across. It may have been taught to you already, which is the particular example of thalidomide. Let me just find the relevant slide here. Right, so despite stringent testing precautions, drugs with harmful side effects can sometimes be passed for general use. Now, thalidomide is a drug that was sold during the 1950s and 1960s to pregnant women, and it was intended to combat morning sickness. Now, later, in 1960 to 1961, it was found to cause abnormalities in fetal development, most visibly causing incomplete limb development, especially if taken during the early stages of pregnancy. So in 1961, Dr. William McBride, an Australian obstetrician, was the first to link thalidomide to birth defects, but the drug was not actually banned until 1962. During the time it was prescribed, around 12,000 children in 46 countries had been affected, of which only 8,000 survived past the first year of life. Most of the survivors are still alive today, nearly all with disabilities caused by the drug. So here we have an example of a drug which was passed through the early stages of drug development and went into general prescription and use in the population, but it was only later discovered to have serious side effects, which eventually led to the banning of the drug. So most UK survivors of thalidomide are now in their early 50s. So thalidomide is now being prescribed for people suffering with painful mouth ulcers, where it's been found to clear up the symptoms within a matter of weeks. AIDS sufferers in the US have recently been prescribed thalidomide, where it's been found to alleviate the symptoms of the disease. In third world countries, thalidomide is prescribed for patients suffering from leprosy, as it can reduce or even clear up the symptoms. Now, obviously, these are specific cases in which the prescription of thalidomide continues, but because we do know that for pregnant women it does cause developmental defects and birth abnormalities in the children, its use for treating morning sickness has been discontinued. So thalidomide stops the growth of blood vessels. So the development of many cancers depend on the growth of blood vessels to supply the cancer cells with nutrients and oxygen. Therefore, thalidomide is now prescribed for the treatment of many cancers, as it stops the development of blood vessels and thereby stops cancer. So thalidomide has even been shown to reduce the unpleasant side effects for cancer patients of some chemotherapy treatments. So we see here that despite the fact that thalidomide was identified as being dangerous for use in pregnant women, it continues to be used for treating various other disorders in non-pregnant individuals. Now, another example that's relevant here is statins. Now, I mentioned that statins have been made available widely on the NHS, but they do still cause side effects in some patients. So some of these side effects include headaches, insomnia, liver problems, digestive tract problems like abdominal pain, diarrhea, and sickness. Now, statins can even cause hepatitis, skin rashes, and serious muscle problems as well. Now, statins can be useful for treating cholesterol abnormalities, but again, it's only after the wide-scale use of statins began where these problems became identified. This has caused kind of a curtailment, a reduction in the prescription of statins, and also much closer monitoring of the patients to whom they have been prescribed. So I hope that answers your question. Let's take a look at the next highest voted question here. Okay, it seems like the the next highest rated question across all 5 groups of students is again to review the distinction between non-specific and specific immunity and how this is different from active and passive and naturally acquired versus artificially acquired. Okay, so let me let's just get rid of that for a moment here, and I will explain. So in order to understand the way that immunity is categorized, generally speaking, we divide immunity, which is the ability of an organism to resist infection by pathogens and to destroy abnormal cells. We divide it into the components by which that can happen, or by which the organism ensures that that immunity happens. So immunity is generally divided into specific and non-specific. So this is the way that we categorize the mechanisms of immunity. So again, non-specific are ways in which the body or the immune system kills a broad spectrum of pathogens. In other words, it's not specific. It doesn't specifically target any particular pathogen. Now, in contrast to that, we have specific immunity. Now, non-specific immunity is sometimes called passive immunity. Specific immunity is sometimes called active immunity. So specific immunity is the ways in which the body, in particular the immune system, targets and protects us against particular specific pathogens that it identifies as being present in the body tissues. So these are the two general categories into which the mechanisms of immunity are divided. So once again, non-specific immunity is either the physical and chemical barriers to infection. So in other words, the skin, the tears, the pH of the body tissues, the oxygen levels of the body tissues. Again, certain body tissues are high in oxygen concentration. So for example, the tissues lining the air spaces in the lungs will typically have a much higher concentration of oxygen in the extracellular fluid than perhaps tissues deep within organs, such as the liver, for example, or within muscles. Now, what this means is certain types of pathogens which are obligate aerobes are more likely to invade tissues where they have access to high concentrations of oxygen. So obligate aerobes are organisms which have to respire or prefer to respire aerobically. Obligate anaerobes are organisms such as microorganisms which thrive under conditions of low oxygen. And so having a high oxygen concentration in the tissues lining the lungs and the airways, for example, is itself a barrier which discourages infection by obligate anaerobes. But also pH. So the pH of certain body tissues is going to be a barrier to infection. Obviously, the stomach we've discussed already, but the pH of other body tissues, such as the blood itself, will prevent microorganisms which typically thrive at pH levels outside of that range from colonizing. So we have physical and chemical barriers to infection, but also non-specific immunity. Part of this is the phagocytic activity of certain types of white blood cells, including macrophages, neutrophils, monocytes. Now again, I simplified this somewhat for GCSE consumption, but actually the different types of leukocytes and lymphocytes are very varied and much more complex. So specific immunity or active immunity can be divided either into the humoral response, which is the response whereby B cells produce antibodies, but there's another aspect to the specific immune response which kind of falls a bit outside GCSE level, and this is the cellular response or the cell-mediated response. Now this involves activation of T lymphocytes, antigen presentation by macrophages, but this kind of goes a little bit beyond the level that you need to be familiar with for GCSE. But this is the general categorization of immunity in terms of non-specific immunity or passive immunity and specific immunity or active immunity. Now, specific immunity can be acquired. You can get specific immunity by two different general methods. You can either naturally acquire specific immunity. So an example of that would be you catch a cold from somebody and your immune system then mounts an immune response against the cold, clears up the cold, but you have memory B cells which then remember the antigens from that particular pathogen, so that if you are then infected by the same virus later on, your immune response is much more rapid and much stronger. And this is naturally acquired specific immunity. There's also passively acquired specific immunity or naturally acquired specific immunity, which you can get in a passive way. So you can get that by, for example, receiving antibodies in mother's milk. You can also acquire specific immunity artificially, and this is how vaccination works. By receiving a vaccine artificially, you have specific immunity which you have acquired artificially as a result of receiving the antigens to that particular pathogen and your immune system developing immunological memory against that particular pathogen. And so this diagram shows how immunity is characterized in terms of the mechanisms of immunity. But when you're talking about naturally acquired or artificially acquired immunity, you're talking about the means by which you got your specific immunity. And so if it's naturally acquired, you acquired it through natural mechanisms such as being infected by a particular organism and then developing immunity to it, or you acquired it artificially by, for example, vaccination. So I hope that answers your question. Great. So, all right. So by the looks of it, that answers the questions in the Q&A panels, or the vast majority of them. The other ones appear to be not particularly relevant to the topic at hand. So I'm going to stop there. So to everybody who's in attendance, thank you for attending. I hope that was useful to you. And please do keep the questions coming on the private resources page. I will also upload, as I said before, the resources, the interactive video for this, the slides that you've seen, the mark scheme for the exam style questions. But again, there is also the function for those of you in attendance to go to that page and continue to ask me questions, continue to correspond with me either privately or through the live chat on any of the topics that we've covered or any of the exam questions that you receive. So please do keep those questions coming. All right, guys, thank you very much for attending. I hope that was useful, and I will see you in the next one. Take care.